So the program for this tutorial is I'm going to give you a really short presentation, about 10 minutes, on Assaymetrics and how we got started with fluorescence lifetime, products that we've brought to market, and some of the applications that our users are putting fluorescence lifetime products to right now. And then, most interestingly, Jon Wingfield from AstraZeneca at Alderley Park is going to give us around about a 30-minute presentation on some evaluations that they've done at Alderley Park and AstraZeneca on our Fluospec and PURETIME fluorescence lifetime platform. Okay, so let's kick off then. So the title of the tutorial then is Drug Discovery with Discriminating Fluorescence Lifetime Technologies, which is a bit of a mouthful. I apologize for that, but it does convey the message that fluorescence lifetime is more than just an instrument, it's more than just a set of reagents. You absolutely have to have the two things together. And one of the reasons why fluorescence lifetime has taken maybe 10 years to get seriously off the ground is that there's been a set of interesting dye reagents available from Amersham. There's been an instrument available from TCAM, but never at the same time. And the two sets of things have actually never been put together until recently. Let me give you a little bit of background about our company, Assaymetrics. I founded the company back towards the end of 2004, and we started doing research and development, basically. In 2006, we filed patents on our fluogenic peptides. That's the basis now for our enzyme substrates that we sell under the PURETIME 17 label. In September 2006, we launched our dye reagents to the world at SBS in Seattle, which was really so exciting and so distracting for me that I missed the plane home. I was just so keen to stay there and just soak up the excitement of the atmosphere. May 2007, we started our development of our own fluorescence lifetime plate reader with a few kind of interesting ideas and a little prototype that we had working in a tiny bit of lab space that we borrowed off somebody else. And from that point onwards, life really changed for us as a tiny sort of four-person company in 2010 when an investment consortium headed by Finance Wales stuck a lot of money into the company, and we did some recruiting and we invested heavily in manufacturing, development for manufacture, and we now have the Fluospec FL instrument, which we're now selling. So we launched the instrument in the first quarter last year, the Fluospec FL, and again, was at SBS. So for us as a company, actually, SBS, or SLAS as we are now, has been really a key organization for us. So what are our products then? We sell what we call PURETIME dye reagents, and we characterize these by their fluorescence lifetimes. 1, 3, and 5 are near-infrared emitting dyes, right the way through PURETIME 14, 17, 20, 22 nanoseconds of lifetimes, and PURETIME 325. So that's an ultraviolet excited dye which emits right at the violet end of the spectrum. And that tells you something actually quite interesting about fluorescence lifetime. It's generally true that you don't see really long fluorescence lifetime dyes which emit at long wavelengths. There are good physical reasons for that, but that's just a general kind of truism just to give you a guide. I'm going to spend the rest of my time talking to you about the Fluospec FL instrument, which is a plate reader designed for high-throughput screening, profiling, and also for assay development. So the features of the Fluospec FL are that it has a laser excitation source. So unlike other multi-modality plate readers that many of you will be more accustomed to, this has a laser source which can be focused down to a really tiny spot, which gives you very good spatial resolution and good rejection of the contributions that you can get from the edges of plates, etc. The Fluospec FL is designed to cope with all the standard plate formats now, 96 384s, low volume 384s, and 1536s. John's going to be talking a little bit later on about assays that are formatted into 1536s, and the instrument works just fine in 1536 mode. Time, one of the things that always used to be said about fluorescence lifetime was it's really slow. Really quite interested, the response might be, yeah, really quite interested in fluorescence lifetime. Discrimination advantages sound kind of interesting. But the old story was that it would take minutes and minutes to make a single measurement. That's all changed these days. 200 milliseconds is the standard acquisition time for a well. The Fluospec also gives you data analysis on the fly, so you don't have to wait until the plate's been read to give you information about the fluorescence lifetimes, gives you all that information immediately. And something which is unique to our reader, PureCheck is what we call the feature which detects and then corrects for the presence of interferences in individual assay wells. Those might be interferences due to compound fluorescence, it might be due to fluorescence due to compounds, components of the buffer, or it could be due to absorbing compounds. Or it could be due, for example, if you're doing an IC50 measurement and you're working at the sort of top end of the concentration range, where your compound starts to drop out of solution. Unfortunately, wouldn't it be nice to know that you've got a compound which is starting to drop out of solution? Well, the Fluospec gives you that in the shape of an elastic scattering parameter, which it records simultaneous with the fluorescence lifetime, with the fluorescence intensity, and with a handle on the compound absorption. Okay, so those are the features. What kind of benefits does that really bring? So what we're seeing demonstrated now with the instruments, first of all in big site trials and now in customers' hands, is lower rates of false positives and false negatives when it comes to primary screens. Seeing that really it is actually much faster than they thought it was ever going to be. Quickly reading a 1536 well plate in 12 minutes. And in terms of costs of ownership of a new piece of capital equipment, quite attractive actually. Lowering HTS costs, hit-to-lead profiling costs, basically because you're able to work with miniaturized assays, and also because when you're thinking about discriminating technology, time-resolved fluorescence is I guess the one that springs to mind to most people. Unfortunately, typically the reagent costs are rather high, and this is something that, because our PURETIME floors are small molecules, relatively straightforward to make, are much more attractive from a price point of view. Also, because they're small molecules, it's actually rather easy and straightforward to format assays. So typically, you can expect to experience faster assay development times. In terms of applications, so what can you do with this chemistry and the instrumentation? Well, you can do a range of kinase assays, protease assays. In fact, our early adopter who's kind of blazed the trail for us in terms of fluorescence lifetime is the Novartis group in Basel. And they've now converted more than 100 of their protease profiling assays over to our fluorescence lifetime platform. Binding assays are also possible in the fluorescence lifetime mode, for example, receptor-ligand, protein-protein interactions. And also now we've come to an agreement with Almac, who supply their FlexSite range of reagents and substrates. And in fact, we've demonstrated a range of their assays, particularly in the epigenetic area on the Fluospec, and they will work really nicely. Okay, so almost through the introduction. Let me just explain a little bit about how you do a fluorescence lifetime assay. Well, let's take one step backwards and talk about the assay model that we're going to use for this fluorescence assay. All of you, I guess, will be really familiar with this model where you take a substrate, which is active, a fluorogenic substrate, which is acted upon by an enzyme and is turned into a product. And the product and the substrate have different fluorescence characteristics. So this is the basis for all FRET assays. With fluorescence lifetime, it's exactly the same, apart from the fact that you get additional information. And what we've done here is just to write out in the form of a simple equation the fluorescence time response. And what you see here are these three exponential terms. The first term here in orange, which is the response to the substrate, decaying away. If you look at that exponential function, minus t, the time over tau. Tau is the fluorescence lifetime of the substrate. Together in the assay well, you also have the fluorescence contribution of the product, again decaying away with a time constant minus t over tau. And also, simultaneously, you detect the contribution due to background. And because you detect and measure the contribution due to the background, you have the opportunity to make corrections for background contributions in assays. So this is the main difference, I think, between doing conventional time-resolved FRET and doing genuine fluorescence lifetime, the period over which you measure the fluorescence intensity. So you make no assumptions that after whatever it is, after 60 microseconds or 60 milliseconds, that all of your competing fluorescence has disappeared. With a fluorescence lifetime assay, you measure it all the time, and then you attempt to correct for it. Okay, this is my last slide, the busiest of the slides, but hang in there with me. So what we have plotted here is a decay curve. It doesn't look like a curve, it doesn't look like a set of curves really, more like a set of straight lines, and that's because we've plotted the fluorescence intensity here on a log scale up the y-axis and time along the x-axis. What we have is the blue line here, which is the fluorescence intensity of the product. So this is actually a product substrate, and it's around about 100 nanomolar. We have the orange line, again on the log scale, we get a nice straight line. That's the substrate, typically a shorter fluorescence lifetime. And then we have the interfering dye compound, which we chose. We thought to ourselves, well, what would be the worst possible interfering compound that you could come across in a screen, or if you wanted to measure its dose response? Well, in fact, looking at the fluorescence characteristics of our PURETIME 14 dye, the absolutely worst one would be coumarin, which excites exactly the same wavelengths as our PURETIME dye and emits exactly the same wavelength. Happily, it has a very much shorter fluorescence lifetime. And you can see that by the steepness of the slope of that green line that you have there. The other two lines, which I just need to explain then, are the red line and the violet line. The red one is a mixture of the product peptide and the dye, and the purple one there is the substrate and the dye. So you can see actually that those two curves that sit between the other three look like they're superpositions of a pair of straight lines in each case. And in fact, that's exactly what they are. Now, you can use that information. One of the simplest ways in which you can apply fluorescence lifetime discrimination is to do something called tail fitting. It involves a little bit of sacrifice of a lot of the signal, but it gives you a signal which is very much more robust and doesn't contain this interfering short fluorescence lifetime signal. So if you look at the slopes of the red line, and which is the product, and you can put product in the dye, and you compare that with the slope of the blue line, which is just the product, to the right of that dashed line, you can see the slope is actually identical, isn't it? And the same is true of the substrate and the dye, the purple line, and the substrate, which is the orange one. So you can see there, after, to the right of that dashed line, the slopes of the lines are identical. So this is just a simple explanation of how fluorescence lifetime achieves discrimination against fluorescent impurities, compound fluorescence in real life. And Jon's going to tell us about an evaluation of the instrument and the dye system that they carried out at Alderley Park. Very pleased that Jon's agreed to come and tell us about his experiences of making this evaluation. Jon is an associate principal scientist at AstraZeneca Alderley Park and has been doing high-throughput screening, really interested in novel technologies for screening and profiling for many years. So Jon, I'll hand it to you. Thanks, Jon.
Thanks very much. So I should point out that in no part have I been paid in any way, shape or form for this presentation, so Assaymetrics haven't contributed to the flight or anything like that. So everything I'm going to say is completely impartial, and if you have any questions, I will give you my impartial perspective on that. Again, I don't necessarily want you to leave here thinking that I'm giving you an official AstraZeneca view of any of this either. So let me make that clear right from the start. So as Pierre said, we've been working with fluorescence lifetime, I guess, probably for about 18 months. And we've worked with Assaymetrics, and we've also worked with Almac and looked at some of the other readers that are available on the marketplace today as well. And I think for us, one of the key things we were really interested to see was the evolution of a 384 into a 1536 plate reader. From our perspective, it's really important that we've got something that we can use for an HTS application, but also for a downstream processing secondary screening perspective as well. So within AstraZeneca at Alderley Park, we are now formed into one screening organisation. So both the high-throughput screening and the secondary screening group are all managed by the same line, and for the first time, we're really able to influence right the way across the whole range of screening, right the way through from primary screening right the way through to late-stage LO screening and preclinical screening. So it's important that we have a strategy that's able to meet the needs and changing throughput requirements right the way across that pipeline. So the assay I'm going to talk about today was some work we did on a tyrosine kinase. Now, where I work at Alderley Park, we have a very large oncology group, and for obvious reasons, oncology have been involved with kinase screening for quite some time. So over the last 10 years that I've been at AstraZeneca, I've done an awful lot of kinase screening. And as I'm sure you're aware, there are multiple methodologies that you could use for those kinases. And actually, I've worked on all of these. So when I first came to AstraZeneca, we ran a kinase screening panel that was made up roughly 50-50 of tyrosine kinases and serine threonine kinases. The tyrosines, we used ELISA methodologies to look for inhibitors, and for the serine threonine kinases, we used the classical radioactive filter capture type assays. But the sorts of throughputs we were getting to really became quite problematic. We were generating an awful lot of radioactive waste, which isn't pleasant at the best of times. ELISAs, of course, from a tyrosine kinase perspective, there are lots of antibodies available, PY20, those sorts of things. It works. It does work pretty well. The issue really is around the number of washing steps. And of course, because you've got washing steps, 1536 becomes a little more troublesome. So most of the ELISAs that we've ever run at Alderley Park have always been in 384 format. So we then progressed to look at assays in the sort of more homogeneous formats, HTRF, alpha screen, and of course they're great because you can work with them in 1536 formats, so you get the throughput that you need. However, one of the problems you've got with them is that they're pretty much stopped assays, so they're great for an HTS and they're great for doing IC50 follow-up work, but if you want to do kinetics, then that could be a real problem for you. As Pierre kind of alluded to in his talk before I started, there are some issues. I mean, the europium isn't cheap, all right? You don't get much of it out of the earth. That's why it's a rare earth metal, right? So the reagents can be quite pricey for those types of assays. Again, alpha screen. We've worked with many alpha screens, and if you want to come up and talk to me afterwards about what I think about alpha screen, I'm more than happy to tell you. We don't run as many as we used to, let's put it out. Again, the big selling feature with these sorts of proximity type assays is that, of course, you've got time-resolved components in there, so you can get fewer artifacts. Well, in our hands, you still have artifacts. It's a homogeneous assay format, so there are going to be artifacts. You just have to accept that. The complexity of these assay formats, again, can actually amplify the number of artifacts that you're going to see as well. Latterly, we've run an awful lot of the caliper electro mobility shift type assays, the lab chip assays for tyrosine kinases. And again, in our hands, that works very well. That's a semi-homogeneous assay format, so you mix the reagents in a well, and then once the assay is stopped, you then sip the reagents out and do a separation, basically via capillary electrophoresis. So that's great. We don't see too many artifacts using that technology. That works well. People understand it. The separation works very well. And there's no requirement for antibodies. So it's not quite label-free. So you still have to have a fluor on there, but it's getting quite close to being label-free. The downsides, of course, are that you have to buy the chips, and the chips are not cheap. So in England, we have a phrase that's cheap as chips, but not these chips. These chips are actually quite pricey. So So the throughput costs can be a bit prohibitive from an HTS perspective. So I'll not go into too much detail here because obviously Pierre knows a lot more about this than I do, and he's probably done a better job of presenting the fluorescence lifetime theory of it than I'm going to be able to do. So, but suffice to say that, of course, all fluorescence dyes have a lifetime, and what you really want is something with a longer lifetime. So GFP, for example, I think has a lifetime in the sort of 1, 2, 3 nanosecond range. And really what you're looking for is something with a lot longer lifetime if it's going to be discriminatory. The thing that kind of attracted us to FLT in the first place was, and one of the reasons we wanted to do the evaluation was really to test out whether or not there were fewer artifacts than with other fluorescence-based technologies. Because it's independent of the excitation intensity and that the system isn't dependent on the dye concentration, then it makes it kind of volume independent. So it's relatively easy to scale from 384, 96, 1536. All of that stuff kind of works really nicely here. Everything scales down really well. You don't have to worry about titering or retitrating an antibody or a bead concentration. And of course, specifically in the case of the kinase, the SARC tyrosine kinase that I'm going to talk about now, the lifetime of the fluorescence can be impacted by the local microenvironment, in that you can have a local quencher, you can relieve that quench, and then you can see a change in the lifetime as a result of that. So this very briefly is something you can get from the Assaymetrics website, which of course is where I stole this from. I'm more than happy to pinch other people's ideas. As you can see here, we've got a peptide substrate with a PURETIME 17 dye attached to that. And in the native form, when it's unprocessed, it has a relatively short lifetime. To give you an example, typically if you put this dye into a well and read it on a plate, you'd be looking at around about a 13 nanosecond lifetime on that. Of course, when you add the kinase and you come in with the ATP, you phosphorylate that tyrosine, then that changes the microenvironment around the dye, and the lifetime changes significantly to greater than 15 nanoseconds. So we were very keen to see if we could do the assay developments directly in a 1536 volt plate. And some of the error bars on here will obviously tell you that this is pipetted by hand, and it was pipetted by my hand, and I don't get into the lab as much as I used to, so that probably explains why some of those error bars are a little bit larger than we'd like to see. But effectively, this is the typical sort of assay that you would do to look at the protein concentration that's required in your assay and see how that varies, see how the assay readout develops over time. I should say that we've run SARC tyrosine kinase historically within AstraZeneca as not as a project. It's not an active project, which again makes it easy for me to talk about it because it's not a target that we're particularly interested in at the moment. But we run it mainly for selectivity purposes. So it's an assay that we've run historically over the last 10 years. So we have quite a large data set, and that made it attractive from that perspective because we could take compounds that we knew or thought we knew where they would be in terms of having an IC50 value against the tyrosine kinase, and then run it in FLT format and see if the data actually correlates. So we've got a benchmark to run the FLT assay against. Because of that, we knew that the Km for ATP was around about 40 micromolar, so we didn't have to run those sorts of assays in order to do this assay development. And then we worked through the final assay conditions that we were going to need. We wanted to make 2 reagent additions, so there are 2 1.5 microliter reagent additions. One reagent has the enzyme in, one has the substrate in, so it's a fairly simple assay to automate from that perspective. In terms of the amount of enzyme that's going in, we were running at roughly 1 nanomolar enzyme. And it was the same enzyme stock that we purchased from Millipore, so it's commercially available enzyme. It's the same enzyme we would use in the HTRF assay. In terms of the buffer mix, that's absolutely identical to the HTRF assay that we run. So in essence, The bit that we validated here really is to understand what titer of the SARC enzyme we need to put into the assay. And when we went back and checked against what we were running in the HTRF assay, it turned out we were in exactly the same ballpark. So we had confidence the assay was going to run. As I hope I said earlier, but I can't frankly remember, but because it's a kinetic assay, obviously this type of experiment is really easy to do. You set the wells up and you just read the plate every 5 or 10 minutes. So from an assay development perspective, that's really kind of nice. The other type of thing we would like to do with this assay would be the sorts of washout type experiments where we look at on rates and off rates. So again, because it's kinetic, it lends itself very nicely to that. So I just wanted to put in a slide so that I had an opportunity to talk a little bit about the reader. So one of the things we were concerned about was the read time. So we worked with a 384 plate reader, fluorescence lifetime plate reader, and we really wanted to make sure that this 1536 reader could cope with a 1536 read that would be roughly equivalent to the same sort of read times you get out of a Ferrostar or an Envision, you know, a standard optical density type plate reader. And indeed they are. They're very close. So the instrument that we had, we were looking typically at a sort of 15-minute read time for a single fluorescence lifetime reader. And that was really quite reassuring. It's kind of an interesting machine if you get a chance to stop by the booth and have a look at it, because unlike most plate readers that I've dealt with in the past, it has a portrait transfer station on it, rather than a landscape transfer station. And from an integration perspective, that makes it kind of nice because it means you don't have to put the plate down and regrip. If you're picking a plate out of a Lyconic or a Cytomat type carousel or incubator, of course the plate's presented in portrait format. So normally, to put a plate into an Envision, for example, you have to pick a plate with a robotic device handler, put the plate down, pick it up again, having in landscape format. Now, of course, the problem with that is that typically we put our barcodes on the east-west sides of the plate, and if that's the way you're actually holding it with your gripper, then it's kind of tough to read the barcode. So, from that perspective, unlike many of the plate readers that are on the market today, we felt that actually having a portrait orientation on the plate carriages was kind of useful too. The PC ran off of a Windows 7 operating system, and for the duration of the time we had the instrument in the lab, we didn't have any great issues with either the software or the operating system. So it seemed like it was a fairly robust software platform. The operating software itself, again, is pretty straightforward. It's not particularly complicated to set up, and I've got a screenshot on the next slide that will show you roughly what to expect and how to input the data so that the files are generated appropriately. One of the really nice things, and again, Pierre alluded to this in his 10-15 minute talk at the start, was really the additional information you get. You don't just get a fluorescence lifetime read out of the instrument. You get a fluorescence intensity read, and of course the software uses this additional information to really flag up and highlight wells that may be a concern. And that's nice to be able to see, because it gives you a really quick, easy way to identify where you've got a problem, if you've got a problem at all. It's got some chi-square functionality, and again, as Pierre talked about, the elasticity measurements and the pure check really give you added confidence that in the data that's being generated by the instrument. And again, from an output perspective, the data is quite straightforward to output in either a string format, 384, or 1536 string, or whether you want that presented in a more standard plate layout that a lab scientist probably can get their head around a little better than just a long string of numbers. So this is what the software looks like. You can see the light blue patch is the part of the plate that's already been read. And in this case, it's bistrophulating obviously up and down, as opposed to in rows. The wells that have been highlighted in red are the wells where the system has done the analysis, initial analysis, and identified there's a problem. So quite clearly you can see in this case that there are some wells on the plate where there are some issues. In terms of the the information on the panel to the right, this is really where you put in the information about the protocol. The system will give the plate a time, date, stamp, unique identifier. Obviously, if it's got a barcode on the plate and it's integrated with the barcode reader, then that's where you'd expect to find the barcode. And there's a few pieces of information you can put in around the type of model that you want to use to analyze the data, or to check that the fit is appropriate, and obviously an output path for the data files. And really, that's kind of all there is. To it. You get a little graphic at the bottom left-hand corner, which allows you to look at the shape of the lifetime in each well as it's being read. I've got to say, it's kind of tough to spot the ones that are the unusual wells if you just look at that. Maybe if I'd had a little more training, a little more time, I might have been able to do that. But you can see that changing, and you know you can kind of see if you get something that's obviously got a very long lifetime or a really, really high level of fluorescence, endogenous fluorescence, then you can see that changing in that portion of the graphic. So a little bit about the compounds that we put through as a test set. We had 2 test sets of compounds. We picked about 5,000 samples from a kinase library. So we knew there were kinase actives in it, and we tested these at single point, 10 micromolar, as if it was a standard HDS type of run. We also selected about 400 compounds that had been through the HTRF assay with a range of IC50 potencies, and we wanted to test these again in the FLT so we could do a side-by-side comparison. Our typical dose response range that we use at AstraZeneca is a 12-point dose response, and it goes from 100 micromolar down to 0.1 nanomolar. So quite a wide range. Within within the 395 compound test set, we had 178 compounds that we had HTRF IC50 data for, and we'd also knew were spiked into that 5,000 test set single point. And here's the data from the single shot. So we had off-plate controls for this, so we set up a different set of controls on a separate plate for each of the plates that we put through the reader. And these are clustered in there's basically 3 full plates and a part full plate. And really what we were looking for here was to see how many compounds fell outside of the sort of greater than 125% inhibition and less than minus 25% inhibition. The outliers, how many were there really likely to be? And rather strangely and kind of fortunately, there were about 50 in each category. And even I can do the maths on that, so 50 out of 5,000 is fairly straightforward to work out what the rates were. We went back and looked at the intensity, fluorescence intensity figures for the compounds that fell into that upper category, and these are absolutely highly fluorescent compounds. The 50 or so that fell below the minimum cutoff, these were compounds, when you go back and check through, the data analysis kind of suggests that these 50 compounds may have long or extended lifetimes. We may need to, I guess we could have probably cleaned this up a little bit if we'd changed the parameters around some of the additional analysis tools that are available within the software. So we don't run samples in duplicate at AstraZeneca. The reason for this is because we believe a better and more appropriate approach is to actually take the compounds and run them in 2 completely independent, separate tests. We do the compound dosing with an acoustic dispenser, so we're pretty confident that we've got a lot of accuracy in the way we dispense the compound. So what you're looking at here are IC50 curves generated from 2 independent tests. So the compound plates are produced twice, all the buffers are made up twice, and everything is tested completely independently. So nothing is run at the same time. And you can see from this that the data correlates between the two runs pretty well. So the red line is the XY line, and the black line is the best fit line. So the two don't quite sit right on top of each other. But that probably is a good thing, because then you think I just fixed the data right. So the fact that they're slightly off center is probably a good indication that we were doing quite well. 1536 dispensing in this sort of format, we use a bioreactor, and that's really because we like the precision of that instrument in this sort of dispensing range. There's 120 compounds per plate, and that gives us enough room to put on-plate controls. And we're also quite careful in a secondary screening environment that we use randomized controls, so we don't put all the controls in one column or in rows. They're actually scattered across the plate in different positions, so that we don't run into issues with edge effects. So the Z prime for this assay it's reasonable, it's okay. We've run biochemical assays in the past, and you can get better Z primes than the one we've got here. But we were reasonably happy with this in this format. So this is now looking at the 178 compounds that we had IC50 data for, and single shot data. So the single shot data that you see presented up the side of the graph there is from the single point percentage inhibition data when it was tested, when these compounds were tested at a single point. Across the bottom, we've got the HTRF assay PIC50 values that were generated, and really the key for us on this was to look at, at 50% inhibition, okay, what sort of IC50 would you expect to get? Right? Now, 50% inhibition, we started with a 10 micromolar compound concentration, so you would expect it to be around about in the sort of micromolar range, and that's exactly what you see here. So that gave us some confidence in terms of that correlation there. There are obviously a couple of outliers, and again, when you go back and look at those two at the top left-hand corner, they are a couple of the very highly fluorescent compounds, and that's why they sit in that outlying region. So we then went back and looked at the 394 compounds and said, okay, what does their historical data set look like? And they compare reasonably well almost in terms of a ranking. So the HTRF data and the FLT data correlates reasonably well. However, there does appear to be some sort of potency shift going on here, because they don't sit on that red line. The more potent the compound gets, the further away from the red line it moves as well. So we don't really understand quite what's going on here. We've got some plans of what we want to do to try and work out exactly what's happening, and which of the IC50s is the correct IC50, if there is such a thing. So to summarize, the reader itself we found to be very simple, very straightforward. The data acquisition times were exactly where we would want them to be, and to make the instrument comparable with the other sort of spectrometers that are available on the market for reading in 1536 formats. The fact that the assay formats allows us to run things in a kinetic mode, we felt was really useful from an assay development perspective, and gave us some options that we could do downstream to look at mode of action type assays if we did find inhibitors. The artifact rates from the fairly small subset that we tested, as I say, about 50 compounds were showing up as being highly fluorescent. About 50 compounds out of those 5,000 looked like they had lengthy lifetimes. The run-to-run variability looked to be very, very good. We didn't really have any great issues with that. And that's something, to be fair, that we found when we've done other fluorescence lifetime work with other readers. We don't see too much run-to-run variability, whether it's in a tyrosine kinase model or whether we're in a serine 3 kinase assay either. And as you can see from the previous slide, the IC50s that were calculated in the FLT assay were comparable to these sort of IC50s that you would get from the HTRF assay, although obviously there was that potency shift. But if what you're looking for is a way of ranking your compounds, then we feel that they would probably rank very similarly in either assay format. And clearly from our perspective, in terms of trying to get a better handle on exactly what's happening here, it's going to be really important to understand the differences between the two assays. And at the moment, obviously, with the exception of the detection technology, the only real difference is, we suspect, is really around the substrate that's being used. As I mentioned earlier, the reagents that are going into the assay are pretty much the same, whether it's an HTRF assay or whether it's the FLT assay. The difference is probably going to be substrate. We understand what the substrate is from an FLT perspective, but from at the moment it's not clear to us exactly what the CysBio guys are using as a substrate in the HTRF assay. So when we've got some handle on that, we'll be able to do some mass spec analysis, for example, where we can take the HTRF assay and maybe the FLT assay, run them, but rather than using the fluorescence detection end, we'll just put them straight down a mass spec and see what the relative proportions of product and substrate really are, and calculate the IC50s that way. And that should then give us a reasonably good handle on say, where that variability is actually coming from. So that's kind of what we want to do next, to really bring us to a conclusion really around that evaluation. So I think that's the last slide. Yep. Joe, if you've got any questions that you want to ask, either myself or Pierre.
Great, thanks.